pleased to welcome you to today's PGC Worldwide Lab meeting. For the next hour, you're going to hear about the exciting research which is being done in Africa today, which is also kind of important. Although we're now in an era where genetic studies are yielding unprecedented insights into the biological mechanisms of psychiatric risk, the absence of globally diverse dataset continues to hinder progress and really threatens to worsen health disparities. And this is because the lack of diversity in genomic studies restricts the accurate translation of genetic research into clinical practice and public health policy. It can also lead to inaccurate risk assessments and a lack of interventions for understudied groups. Genomic studies in psychiatry are increasingly prioritizing the inclusion of diverse population groups, and studies conducted in Africa are certainly leading the way in these efforts. So today's Worldwide Lab is going to showcase just some of the fantastic work that is going on the continent, and we have some fantastic talks, which I know you're going to enjoy. If you have any questions during this next sequence of talks, please feel free to post them in the chat and we'll have a Q&A session at the end. So without further delay, I'm going to hand over to our first speaker, who is Dickens Akena in Uganda.
All right, so thank you so much, Conrad, for this opportunity, and I hope everybody is okay, whether it's morning or evening or afternoon, wherever it is. I guess I'll share my screen. So as a way of introduction, my name is Dickens Akena, and I'm a psychiatrist based in Uganda at the Makerere College, at the Makerere University School of Medicine, the College of Health Sciences. Okay, so I'll be talking to you about our work. Just hold on a minute, 1 second. Okay, so the title of my talk is the neuropsychiatric genetics of African populations, the psychosis component of the study. And then I'll also talk about Jinga shortly thereafter. These slides were prepared by myself and Anne Stevenson as well. Okay, so let's just mute this. Sorry. Okay, put this here. All right, so that would be the outline of my presentation. I hope everybody's hearing me well. So we'll talk about the NeuroGAP psychosis project, the background, the recruitment goals, and then the data generation. Then we'll talk about the phenotyping, and then the Global Initiative for Neuropsychiatric Genetics in Education Research, which is JINGA. And I'm happy to see Merwe here, who is a JINGA fellow. So this is the NeuroGAP psychosis team. And in 2021, we won the Pamela Sklar Innovation Award. So the PIs are from left to right, top to bottom. Myself, Dickens Akena, from Uganda, Lokoya Tuli, who is from Kenya, Lori Chibnik, and Bizugelaye, who are from the Harvard School of Public Health and the co-directors of JINGA, and then Simon Kariki, who is also from Kenya and was based at the Kilifi Center. And below is the Professor Karestan Kunen, who is the overall PI of this project. Charles Newton is based in Kilifi in Kenya. Dan Stein at the UCT in Cape Town. Solomon Tefara in Addis Ababa, and Zuki Zingela in the University of Walter Sisulu, that's in South Africa. Okay, so the NeuroGAP psychosis was conducted in 4 different countries at multiple sites within these countries, as you can see, in Ethiopia, in Addis Ababa, and all the studies basically were conducted within the framework of universities, of the biggest universities in those countries. So Addis Ababa University basically was a site of data collection in Kenya, in Ethiopia, and in Kenya, data was collected from Western Kenya in Eldoret, and then in Eastern Kenya in Kilifi. Then the lead university was Moi University, and then the Kemri Wellcome Trust collaboration. In Uganda, there were 4 different sites scattered in the northern part and in the southern part and in the southwestern part. And then we also see lots of diversity in South Africa, from the Western Cape, where there's Cape Town, the little tiny dot on the extreme left of your screen, and then in the Eastern Cape as well as some parts of KZN or KwaZulu-Natal. Okay, so what happened? We ultimately collected, I need to pause. Yeah, okay, great. So originally we wanted to collect 34,000 samples, but we ended up collecting 43,000 samples. And this was the original sample size calculation. But thanks to the Stanley Center and an NIH grant, we were able to increase our sample size to 43,000 people, or just after 43. And then this recruitment was completed in March of 2023. The senior program manager, Ms. Haroki Stroud, there is on the left, with Solomon Tefara and Malcolm, who were basically cutting the cake at the celebration of the 43rd, almost 43,000 samples. So how did we increase this? We had funding from the Stanley Center, our own RO1 grant, and then the Puma's U01 grant as well. Okay. So what happened? Our first 960 NeuroGAP psychosis samples were genotyped by Elizabeth Atkinson, who was at the Broad Institute then, and she has since moved. And basically, she was able to demonstrate huge amounts of variability, genetic diversity, within similar populations within the same population in the East African region. This paper was published in Nature, and basically these were the key findings about the immense genetic and linguistic diversity from these study samples. The study sample was rather small, but we were still able to quickly figure out these kinds of variations. Okay, so then right now, Alicia Martin was also able to demonstrate that low coverage sequencing was a cost-effective method of detecting variations in underrepresented populations. And again, these findings were published in the year 2021. So again, the experts in genetics should be able to understand what Alicia Martin was doing here. All right, and basically this is the publication, and these are the authors. All right, so what's happening? The sequencing is ongoing, and then there's a blended genome exome technique that's being used, the BGE, to do the sequencing of our DNA, and then it's enabling us to identify common variants in these populations. And then already pilot work has been done in underrepresented populations. And because we had such a huge sample size, we really needed to get cost-effective means of analyzing this data. Okay, so that's basically the genotype. So what about the phenotype? What exactly did we do? So this is basically our study flow. So there were cases. So a case was somebody who had psychosis, and then controls was people who did not have psychosis, but all of them were recruited from similar population, and they were matched for gender and age. So a typical example would be that in a mental health hospital, a case would be somebody with psychosis, which was either schizophrenia or bipolar affective disorder. And then the controls would be a participant or a patient who had come to seek general medical health care from the hospital. And maybe this person had malaria or had a toothache or something like that. Okay, so informed consent was obtained from all participants. And first, the participants had to understand the consenting process. And then after that, we assessed whether they had fully understood the consenting process. And then we used the University of Berkeley. We used the UBAC tool, which was supposed to measure the ability of participants to consent. And then once they had scored a certain figure, which was greater than 14.5, then they basically were enrolled in the study. the findings from the preliminary findings from the UBAC have been presented at another setting in the psychiatrics in the ancestral populations network meeting at the NIH. So then participants provided 2 mL of saliva in an origin kit, and then it's from that saliva that DNA was extracted. So the questionnaire basically included demographics, and then the MINI, which was the tool that was used to make a diagnosis of mental illness in cases only, and then the psychosis screening questionnaire and the Kessler-10 in only controls. And then we also administered the life events checklist, the assist to measure substance use, and then the CD to measure presence of physical illness in all participants. We also took measures of blood pressure, height, weight, and then the heart rate. And then we reviewed the participants' charts for previous admissions, whether they had whatever medicines they were taking. Okay. So basically, this is what our application looked like, and the MINI was basically collected in the MINI app, and then the REDCap was used to collect all the other data. Okay. So what were the goals of so we have a phenotype working group, sorry, and the goals basically is to analyze and publish the phenotypic data. Usually all this work is first authored by early career scientists in Africa, and the output is so far 4 papers and 10 papers, sorry, and then 4 of them are in publication. And we can see that from Kenya, we have Lynette Ongeri, we have, sorry, I'm blanking out on her name a little bit, but we have, well, somebody in the audience could help me. But yes, we have Lynette, then we have Edith Koba. Edith Koba is basically the co-PI in Kenya. And then the South African team, again, I think Mary would help me with the names here, because I think I forgot some of the names. Then we have Melkam, we have Barcott, and we have Engida from South Africa, sorry, from Ethiopia. And then from Uganda, we had our own fellows, junior faculty who were involved in writing up this paper, Claire Kwagala, Molly Naisanga, and Zahara Morai. Zahara is Tanzanian, but she was studying in Uganda at that point. And then the people who were helping our colleagues to write up these things included Bizugelaye, who is basically the director, one of the directors of the Jinga Fellowship, Amantia, Anne Stevenson, to whom I give credit for these slides, as well as Christina Kote, who is a clinical psychologist. All right. Oh, wow. Okay. Thank you. So the names are here. Yeah. So the second person is Mary Bita from Kenya, and then Florence Jaguga. I've already mentioned Lynette Ongeri and Edith Koba. All right. So the South African team had Jacob Hoffman and then Yanga Thungana. All right. So what's going on with the validity and reliability study? So we got some funding to basically validate the MINI against the SCID, which is a gold standard, and we conducted a study in 332 participants in Uganda and the same number in Ethiopia. Okay, so the intention was to provide a gold standard comparison of the MINI to the SCID, and then so what happened in this, there were 314 cases and controls, and then an inter-rater reliability was conducted in approximately 100 cases and controls. And the MINI was validated against the SCID in the languages of Kiswahili, Amharic, and Luganda. That is in Kenya, Ethiopia, and Uganda between the periods of May 2021 and September 2022. So now we talk about the Global Initiative for Neuropsychiatric Genetics in Education Research. So why did we have the JINGA? I'll spend maybe just 1 minute to talk about the JINGA. So the reason we had this was over the years, a number of colleagues from the Western Hemisphere have, or across the Atlantic or whatever it is, or the Northern Hemisphere, basically have visited the African continent, collected data gone and basically left the Africans in the same way that they found them, or even worse. So part of the NeuroGAP psychosis and NeuroDev interventions was to make sure that everybody, well, not everybody, but a huge number of the people from the study sites were capable of analyzing this data and making heads and tails out of it. And therefore, the GINGA program was meant to develop capacity for each and every member of the country. So PIs had to give out names of people who would then be able to analyze the data. So in each of the 4 countries, Uganda, Kenya, Ethiopia, and South Africa, there is some capacity that has been developed for the individuals from those countries to be able to make heads and tails of the data that has been collected. So we are extremely proud of whatever it is that has been done in this area. Okay, so there was a first cohort of GINGA fellows, which had 17 fellows. And then now there is a second cohort, and we have Patricia Kipkemoy, Mary Mafford, and Megan Campbell, who are from South Africa. I've seen Mary here. And then we have Raymond Odohonyero and Anita Rinda from Uganda, and then Barcode Samebo and Engida Gebiso from Ethiopia. Okay, so how did this happen? So initially in 2020, we started off with trainings, classroom workshops, and then in 2021, we had the research methods training in Makerere University, and all through from 2020 till now, there are virtual classrooms that are ongoing. In 2022, there was a bioinformatics training in Cape Town, and then 2023, there is a training at the AAU that happened at the AAU. And then there was a workshop in Boston that happened between, I don't recall, but I think sometime last year in 2022. Then this year, we are hoping that our fellows will be able to attend the WCPG in Montreal. And then hopefully at the end of all this, our fellows are able to submit K43 grants from which they can start to become independent researchers. One of the fellows, Dr. Alan Kalunji, from the first cohort, is already a recipient of an important grant. So who are the people who are behind this? There are many, many more people whose names are not here, but it's worth mentioning the names of Karisan Kunen, who is the overall PI of this, and then Bizugelai and Lori Chibnik. And then the different PIs from the different countries whose names you've seen: Solomon Tefara, myself, Dan Stein, Okewole, and Simon Kariki. So thank you very much.
Thanks very much, Akena. The next speaker is Celia.
Thanks, Akena. Can you stop share? There you go. Okay. All right, good morning, afternoon, or evening, everyone. Thank you so much to Conrad for inviting me to come and give a talk today about the NeuroDev project. I'm the research director for NeuroDev, and the PIs are Amina Abubakar from Kenya, Kirsty Donald from South Africa, Charles Newton, also from Kenya and Oxford, and Elise Robinson from the Broad Institute. So NeuroDev is a multifaceted study with many different moving parts, but the primary aim of NeuroDev is to recruit children between the ages of 2 and 17 years with the diagnosis of a neurodevelopmental disorder. And so this includes intellectual disability or developmental delay, autism, ADHD, and/or a language disorder. And we aim to perform genotyping and exome sequencing on 1,880 cases, the same number of age and ancestry matched controls. And we have a nested family design, which includes the recruitment of parent controls and trio families. NeuroDev also includes the characterization of the phenotypic spectrum of neurodevelopmental variation in children ascertained for NDDs, and this is done through a range of assessment tools and deep phenotyping methods. And then what we aim to do is using the genetic and phenotypic material to consider genotype to phenotype relationships in children from this diverse population group. And then in addition to the accumulation and analysis of genetic and phenotypic information, we also collaborate with the NIMH with biobank, so we can store DNA and cell lines. Then we have an extensive list of shared resources that we will be providing our data to. And then lastly, we have a return of results component where we can return findings, pathogenic or likely pathogenic solved cases with participants of the study. So currently we have 3 recruitment sites in Africa. These include our first site in Cape Town, South Africa, which is led by Kirsty Donald. Our second site in the Kilifi County in Kenya, led by Charles Newton and Amina Abubakar. And more recently, we opened a site in Nairobi in Kenya, and this is led by Amina Abubakar. And our samples from all 3 of these sites are shipped to the NIMH biorepository at Rutgers University called Sampled, where DNA extraction occurs. And then the DNA is shipped up to Boston here at the Broad Institute, the genomics platform where the DNA is genotyped and sequenced. And the site in Boston is led by Elise Robinson. And here we oversee all aspects of data management, protocol alignment, and general study management as well. Our recruitment numbers for NeuroDev are updated daily through our NeuroDev website. And the image over here allows you to track the total number of participants, cases, controls, or parents, and you can also subdivide it by each site. And as you can see, during the COVID pandemic, of course, our recruitment was halted for a while, but we're really proud of our teams because the rate has since picked up to a rate akin to pre-pandemic levels. Just to briefly take you through where we are in terms of our targets, we've sort of subdivided our numbers in terms of different waves. So we had an initial pilot wave, which included just trio families, and which I'll be speaking more on results of in a little bit. Then we had our wave 1 samples, which were all the samples recruited up and until March of 2020. And then lastly, we have our wave 2 samples, which is all the remainder samples that until we reach our target. And in South Africa, we're very proud of the team. We're almost complete with recruitment. We've done about 96% of our cases, and we aim to recruit actually by the end of this week. In Kenya, we've recruited about 56% of our target for our cases, and we are well on track to complete the recruitment in our estimated June 2025 date. Then in terms of the phenotyping, as I mentioned, we deeply phenotype all of our participants through a range of different assessment tools. And these tools include measurements for child development, cognition, autistic features, child behavior, ADHD using the SNAP, different medical aspects such as neurology, growth, and motor skills. We also look at socioeconomic status and demographic features. And we have an option for participants to consent to photographs so that we're able to assess child dysmorphologies. And this summary page is also available on our website, and we are you're able to click on each of the tools to download them as well. So to go over a little bit of our phenotype properties in more detail, a total of 211 participants in Kenya were collected for wave 1 and 410 from South Africa, breaking down into 206 probands, and 99 of these were initially sequenced as part of our trio pilot study. Of the 92 cases, 45% were male with a mean age of 9.6, and 219 cases, we had a 71% male ratio with a mean age of 7.4. Figure C is just highlighting again the phenotyping measures that we did. And in figure D, we're very proud of our consent rates for the photographing and cell lines as well. And they're remarkably high, which is going to be extraordinarily useful moving forward. And it also just shows one of the tools called the Ravens or the Multino, which assesses child cognition. Then to underline a few case characteristics, in graph E, you can see our breakdown of case diagnosis, with the majority of cases having both autism and global developmental delay or intellectual disability, and then also some having just global developmental delay and some having just autism. And then we also decided to compare our NeuroDev cases, a subsection of cases, with a different study called the Simon Simplex collection. And we compared this in terms of co-occurring adverse neurodevelopmental outcomes. So what this is showing you is that the NeuroDev study had a lot more cases with more co-occurring adverse outcomes compared to the Simon Simplex collection. So to go into a bit more detail about the exome sequence trios, these were analyzed by the team members at the Center for Mendelian Genomics. And from our analysis of the pilot, we found a high rate of solved cases where we found a pathogenic variant in a known gene associated with a neurodevelopmental disorder. And this high rate was found in both South African and Kenyan trios. We also observed a total of 9 cases with variants in novel candidate genes. So basically what we did is, following analysis, if we found a variant of sort of unknown significance that was on the edge of qualifying for pathogenicity, we would submit this variant and its associated phenotype to a tool called the Matchmaker Exchange platform. And Matchmaker works on this premise that is, for rare diseases, there's not often a clear etiology after exome sequencing. But if you can match other cases around the globe that have variants in the same gene and with overlapping similar phenotypes, it could ultimately provide evidence to shift this novel gene status from candidate to causative. So just our 99 trios, we matched 9 cases through Matchmaker, 4 of which have already been published in case series papers. And then I wanted to highlight that our trio analysis is on medRxiv for those of you interested in reading it. Patricia Kipkumoy is the first author, and we're excited that it has been accepted at Niran and should be published within the next month. I also wanted to briefly mention that our additional Wave 1 samples have completed genotyping and sequencing, and we've just completed our analysis for all of these samples. So this is going to be published at a later date, but we're excited to present that we found a really rich, large number of genetic variants, particularly cases with pathogenic or likely pathogenic variants, as well as these sort of variants of unknown significance that we've submitted to Matchmaker, and we've gotten a large number of matches here as well. And then lastly, I also want to highlight the extensive phenotype data that we generate and how this data is being utilized through a multitude of studies led by young African researchers. So the first study specifically looked at construct validity of the SNAP assessment, which measures ADHD, and this was done by Michal Zief in South Africa. Secondly, our PhD student, Patricia, who's just published our trio paper, she's also working on a number of different phenotypic analyses with our NeuroDev data. We have another PhD student in South Africa, Claire, who's looking at sex differences with our NeuroDev data. And then Bjorn looking at social determinants and also cognition with the UBAC scores, as Akena described. And then we have Dorothy from Kenya, who's looking at, around the ethics of community engagement and recruitment of cell lines from and adult participation from our cohort. For data sharing, all of our trio pilot exomes are available to be downloaded from the Anvil platform. You need to go through DBGAP to access this. We've also uploaded all of our findings to ClinVar, and we are sharing all of our control samples with the NOMAD database. So we've shared a number of child controls from Wave 1 already, and we will continue to share with NOMAD as they release new versions. And there's other databases as well that we're sharing. Face2Gene, we will be sharing our photographs so we can improve the algorithm that clinicians use to diagnose dysmorphologies in African populations. There's also the PGC that we will be sharing our genotype data to be included in the meta-analysis of the GWASs for autism. And then what we're still busy with, we're going to, as I mentioned, complete our recruitment in South Africa this month and in Kenya in June of 2025. For data analysis, we're excited that we've finished our data analysis of Wave 1, but we are still going to continue analyzing this data to look at de novo rates and population-specific changes. And we are also working with NeuroGene, which is the Global Initiative in Neuropsychiatric Ethics, to better understand the ethical implications behind cell line recruitment in our Kenyan populations, and considering how we can start converting the cell lines that we have recruited into induced pluripotent stem cells as well. And then we will continue returning results. We're excited that we've already returned about 27 results from the genetic findings I mentioned back to families. And our genetic counselor in South Africa, Zandri Brewer, will also be writing a paper on the qualitative findings of this action. So that was everything from NeuroDev. Short and sweet, hopefully enough time for Niran and Conrad to do their talks, but we just want to acknowledge our remarkable team and all the work that they've done for the last 5 or 6 years since NeuroDev has been ongoing. We really have lovely people that we work with. It makes it such a pleasure to work. And we'd also like to acknowledge our funding bodies. So the Stanley Center for Psychiatric Research, Safari and the Simons Foundation, and also NIMH and NICHD. Thank you.
Thanks very much, Celia. Great talk. So our next speaker is Niran.
Okay. Can you see my slides?
Yes.
Awesome. So hello, everyone. A very pleasant day to you, wherever on the planet you're listening. So I'll be talking about a relatively new project called DepGenAfrica. That's short for Depression Genetics in Africa. Now, by way of background, we know that major depressive disorder was ranked as the 3rd cause of disease burden worldwide in 2008 by the World Health Organization. And this is projected to increase to a ranking of 1st by 2030. Now, most GWAS for depression have been performed in populations of European origin, some in East Asian populations, and to the best of our knowledge, there are no studies in Africa. We nevertheless know that there's need for large cohorts to study the genetic architecture of depression in African populations, keeping in mind diversity and population structure. These cannot be extrapolated from non-African or diaspora data. It's a short presentation. I'm not going to have time to talk about the epidemiology of psychiatric disorders, specifically depression in African populations, but there are a couple of really good reviews that you might want to have a look at. So it is against this backdrop that the DepGenAfrica project was conceptualized. This picture is from the inaugural meeting in October 2022 in Johannesburg. The study sites include Nigeria, Ethiopia, and Malawi, with technical support from South Africa. So what are the research questions? The 4 key questions that we hope to answer with this project. Firstly, what are the symptoms, the phenotypic characteristics and associations of depression within 3 diverse Western and East Sub-Saharan African countries? Secondly, what is the genetic architecture of depression in Malawi, Ethiopia, and Nigeria? And does it differ from African and European ancestry populations resident in the Americas and Europe? Third, does downstream analysis of genetic data implicate similar biological processes, cells, tissues, and causal risk factors? And are these findings similar to studies of MDD in other climes? And finally, does trans-ancestry GWAS increase the power to find, map, and identify causal genetic variants associated with MDD? So we will be collecting demographic data and data on social indices using the OSS, the life event scale, food security with the HFIAS. Core phenotyping will be done with the MINI, PHQ-9, GAD7, PCLC, and HUDAS 2.0. Other phenotypes of interest will include cognition, physical health, sleep and exercise, substance use. And then for cases only, we would be interested in using the clinical global, we'll be using the clinical global impression, the Morisky for medication adherence, and ASEC for side effects. All participants will have morphometry and phlebotomy done. And of course, there'll be DNA extraction and genotyping and bioinformatic analysis. Now, what is really special about DepGenAfrica is that there are these institutional pairings which leverage existing collaborations that predated this study. So within the hub, there is the University of Edinburgh, represented by the PI, Andrew McIntosh, who co-leads the PGC Depression Working Group. And Edinburgh University is working with Malawi Epidemiology and Intervention Research Unit, and the Kamuzu University of Health Sciences, Malawi, represented by Mia Crampin and Chisomo Msafula. These was an ongoing project with UKRI and Wellcome funding that DepGenAfrica is working with. King's College London is represented by Jerome Breen, who co-leads the Eating Disorders Group and also co-leads the largest study of genetics of anxiety and depression in the UK called the GLAD. So the project has been learning a lot from the experience of the GLAD. And KCL directly partners with Addis Ababa University, with Solomon Tefera, with the wealth of experience that is available there. And then Cardiff University with James Walters, who co-leads the PGC Schizophrenia Working Group, is partnering with Ahmadu Bello University in Nigeria. And then we also have technical pairing with Edinburgh and the University of Witwatersrand, with Michelle Ramsey, who has decades of experience doing genetic studies in Africa and heads the Sidney Perena Institute. So the data processing pathway involves samples, both that combine individual clinical and genetic data, being harmonized from the different sites at the University of Witwatersrand. The combined data sets will have QC done and then be archived at the European Genome Phenome Archive. There'll be two ID-linked data sets, the phenotype data and genetic data. And data sharing subsequently, that includes phenotype and genetic data, will be made available to all DepGenAfrica parties without delay. And then following an embargo period with other bona fide parties. So the project timeline, it's a 5-year project which started in September 2022. There was a kickoff meeting in October 2022. There are annual meetings at the Sidney Perena Institute in Johannesburg. The target is 10,000 participants with major depression. And we will be leveraging population controls from H3 Africa and other projects. Currently, there is a lot of thinking around development of documents, ethics approvals, data and sample collection, data generation, and others. So each of the 3 country sites have their peculiarities. So I've simply given an example of the Nigerian workflow. Essentially, what we are developing is a tiered system where you have individual sites linked to regional hubs, and then with the final coordinating site at Ahmadu Bello University. In terms of general considerations, there's a lot of thinking going on around sample size, genetic diversity across ethnic groups, geographic and environmental differences. Thinking also about what type of genetic data is will best maximize the goals of the project, putting together an analysis team and capacity strengthening. So in all, DepGenAfrica hopes to leverage or will leverage all the opportunities that the African continent provides. Thanks for listening. I will now hand over to Conrad to wrap things up.
Thanks very much, Niran. Great talk. Okay, I'll just share my slides. Okay. So today's Worldwide Lab is being hosted by PGC Africa, which is an initiative which is co-led by Niran, who you just heard from, and myself, and was launched only 9 months ago in last year's World Congress of Psychiatric Genetics meeting in Florence. One thing that you'll have understood from watching these first 3 talks is that African momentum in the field of psychiatric genomics has really been gathering pace long before PGC Africa arrived on the scene. Apart from the initiative that you've heard about today, there are also many other studies, as you can see from this. As you can see from this timeline graphic. So I'm not going to sort of spend much time describing them because of time constraints. But given everything that has already been going on in terms of genetic research in Africa, what more can PGC Africa do to ensure that progress achieved so far continues to be sustainable into the future, especially now that many of these studies that we're looking at have now finished? Well, our mission goal is to facilitate the participation of African populations in genomics research. And within this remit, we are particularly trying to engage African populations that haven't featured in any of the ongoing initiatives that you've been hearing about today. If we take a look at the geographic distribution of where all the studies which have been funded are actually based, we see that there's a lot of overlap in terms of the countries where these studies are taking place, as reflected by these numbers, which correspond to the numbers of studies in any one country. So to us, it's clear that something needs to be done in order to maximize the coherence between research activities on the continent, so that each additional research funding which is acquired is actually going into translating into broader geographic coverage of African diversity. So one thing to point out is that you can see here that there are large swathes where there's no psychiatry-related genomic research whatsoever. And so addressing these disparities in terms of access to research will make it more possible to achieve equitable progress in the delivery of new treatments by maximizing the diversity of African genomes sampled from the continent. And what we're really concerned about and will work hard to prevent is the emergence of new intra-African health disparities in the coming era of genomic medicine. So when we started the working group, we decided on 4 or 5 sub-goals that would potentially bring us, get us close to achieving our overarching aim of facilitating more participation of African populations. Perhaps the most important of these goals is applying for funding, as this is the key to enabling all subsequent goals. And I'll say more about that in a few slides. We also want to put in place ethical frameworks to protect the interests of African researchers and research participants. We want to coordinate recruitment in new areas of the continent, as I've mentioned. We want to cultivate a cohort of PGC analysts based on the African continent, who can then train others and help to ramp up analytical capacity in their local environments. And finally, we want to undertake large-scale analyses of African and other global data. And so the motivation for these goals is really a strong desire amongst African researchers themselves to participate in large-scale genomic studies of African psychiatric populations, and for studies of African populations in general to become more commonplace in the field. And so our priority is to ensure that African researchers are equipped to play leading roles in any research that makes use of African data, and that we also create opportunities to engage with a diverse range of African mental health stakeholders who can help us to shape a communal vision of how progress in genomics research can strengthen the delivery of mental health strategies across Africa. We also want to ensure that any insights from this type of engagement are subsequently used to guide the development of research that addresses the needs of African populations. So here's a quick graphic that demonstrates the progress that's been made in terms of engaging different parts of the continent. We've reached a total of 140 members, and at the country level, our African membership is up to 21. And the total number of countries is 24 if we also include the countries which are part of the African diaspora. So each of these countries that you see here is color-coded to reflect the different geographic parts of Africa, so northeast, south, west, and central. As I mentioned, the focus of our plans is really to expand research activity in countries of North and West Africa. And so accordingly, much of our outreach effort has really been focused on countries in this region. And you can see that much of the growth in membership at the country level has been driven by researchers from these countries. And also just to point out that if there are any African people who are looking at this slide and you don't see your country represented, then the reason is because we haven't been successful in finding people to engage with yet. So maybe you'd consider stepping forward yourself and getting in touch if you want to become part of our friendly community. In terms of the makeup of the group, at the last count, which was towards the end of last year, just under 50% of our membership reported as being senior researchers, and 37% were at the early career stage. A further 11% described themselves as mid-career researchers, and less than 5% at the graduate level. Additionally, 2/3 of respondents are engaged in research that involves data or sample collection activities. A slight caveat to these findings is that they're not based on saturated data. So these results are generated based on a survey in which half of our membership at the time completed. So what's the structure of the working group? So looking at this graphic, we can you can see that there are two separate arms. There's a leadership committee and also a scientific advisory board. In terms of our organizational achievements, we've really created an internal structure that matches the individual disorder groups within the PGC itself. So we have something like 8 out of 12 disorder groups now established, potentially more on the way. So the leadership of each disorder group, as well as being knowledgeable about their respective clinical areas, are also analytically competent, and included among them are many former trainees of the Ginger program that Dickens spoke about, including Celia, our second speaker. So members of the committee have both internal and external facing roles. So internally, they're responsible for leading the Pan-African networks for their individual disorder area. And externally, they serve as ambassadors to the larger PGC disorder groups, which they're affiliated to. The Pan-African disorder groups really provide a way for African researchers working in similar or sort of complementary fields to meet and connect with each other, irrespective of where they are on the continent. Part of the role of the leads is really to facilitate the sharing of information, knowledge, and ideas across the continent. And the hope is really that this will be a good way to seed future collaborations, especially between parts of the continent that don't necessarily have a tradition or of strong research links. And so promoting better connectivity between African researchers is a really important continental need. And so for this reason, we limit access to these networks to researchers who are either based in Africa or are African researchers who are based overseas. with regards to the Scientific Advisory Board, so it's made up of 4 senior researchers, 2 of whom are clinicians, and a clinical geneticist and an epidemiologist. And collectively, the range of high-level experience that they have will ultimately be critical in helping to strengthen our scientific strategy as we look to apply for grants both later in the year and beyond this timeframe. So Tsurai Sidat is a very distinguished and highly decorated clinical academic from South Africa, who we're extremely lucky to have on our team. And Bizu Galaye, so from a PGC Africa perspective, we want to be aspirational in terms of data collection. We want to maximize overlap between studies where possible. There are some really excellent templates that we can learn from regarding how this can be achieved in practice. And most of these templates come from the Ancestral Populations Network, which is a consortium of studies funded by NIH to study the genetics of mental health in diverse populations around the world. As one of the phenotype harmonization leads within the APN, Bizu's expertise is really central to those efforts, and he's without doubt the best person we could possibly have approached to advise us in this important aspect. Similarly, Okewole is he's the country PI for Kenya in NeuroGAP. So he co-leads a working group within the Ancestral Populations Network, which is looking to introduce many innovations in capacity development across a large research network spanning countries in South and East Asia, Africa, and Latin America. So his expertise is especially valuable to us, particularly as one of the main funding mechanisms that we intend to target later this year will carefully scrutinize exactly how the scientific goals that we're will be striving to achieve are going to enable the delivery of capacity in the countries where the research activities will take place. And Zoné Lombard, so as well as being a brilliant clinical geneticist who heads up the DDD Project in South Africa, so she was also co-chair of H3 Africa's steering committee. And H3 Africa, as many of you will know, is or was a pioneering initiative that accelerated human genomics research in Africa. And so Zoné brings great vision and great know-how that we will aim to capitalize on as we try to navigate and find our own place in the landscape of genomic research in Africa. At the International Congress of Human Genetics, which I was fortunate enough to attend earlier this year, Nicola Mulder talked about a follow-up to H3 Africa that would potentially create a series of African Genome Centers of Excellence. And the proposed purpose of these centers would be to leverage genomics-informed precision public health to deliver optimal treatments. But wouldn't it be great if mental health could be adopted as one of the main themes? And so these are the kind of ideas that someone of Zoné's caliber will be potentially able to help us think through. So another arm of the leadership deals with translational science. And the main purpose of this group will be to channel African ingenuity to provide innovative solutions to problems facing psychiatric communities in Africa. And this will probably lie at the intersection of the different disciplines within that group. So before I run out of time, I would like to share an important piece of news relating to funding. So I'm pleased to report that we have had a productive start to the year in terms of grant writing, and we're about to be awarded our first small grant. So the grant is coming from the Stanley Foundation, which is a US-based institution, and this grant award will allow us to take on 2 senior analysts, one in West Africa and one in North Africa on long-term contracts. And these individuals have strong skills in genomics and data science and extensive experience as trainers with H3 Bionet, which is the bioinformatics division of H3 Africa. So we're very encouraged and motivated by the fact that we've been able to attract such high caliber people into psychiatric research. And this is the kind of thing that we plan to do more of. To help our new fellows get acquainted with psychiatric research. So they will be engaged in a multi-year training program where they'll gain hands-on experience of running genome-wide analyses, using psychiatric data, and working with different PGC working groups. An important thing to note is that the fellows will continue to be based in Africa for the duration of their training. But nevertheless, having the opportunity to work across different PGC groups is certainly a great way to facilitate their integration into PGC's sort of frameworks while they themselves prepare to lead important analyses of African data. The other thing to note is that their vast experience as professional trainers in a bioinformatics setting will be very enabling to PGC Africa's goals with respect to training of our membership. And their combined language skills will also enable us to maximize the geographical reach of our training activities to French and Arabic. So in terms of scientific development, scientific goals for this year, so as I mentioned, so we're starting preparations to submit our first major grant later this year. The main elements of this grant will involve new recruitment in uncharacterized parts of North and West Africa, as I've mentioned. In terms of scientific content, we're interested in the overall genetic architecture of severe mental illnesses in Africa. We're thinking about POS portability, we're thinking about admixture, and we're thinking about gene-environment interaction. So acknowledgement slides. And yeah, for more information, please feel free to contact either Niran or I. So before I use my voice, so we've had all 4 talks. So I'd like to thank all of our speakers for a really fantastic set of talks. I'm not sure how we're doing for time, but we're going to move very quickly into Q&A mode. So I'll just have a quick look at the chat to see if there's any questions there. If people have questions, feel free to unmute yourself.
Hi, Conrad, just one question. Someone asked how to join the ADHD working group, so I've just shared my email.
Okay, fantastic. So maybe I'll ask a question. So, and this question is actually for you, Celia. So, so one thing, that we're kind of, grappling with and maybe sort of wondering about is brain drain, and the fact that people that we've identified as there could be future leaders in an African context might suddenly leave or get opportunities elsewhere before they get a chance to pass on the skills that they've acquired. And so I don't know, as someone who's made the transition to the US research system, I wondered whether you had you could say anything from your experiences about the transition to the US system and how that's impacted your sort of career trajectory.
Yeah. I mean, it's tricky. My situation, I think it's always different for every individual. My situation was there just weren't any job opportunities in South Africa. And when job opportunities did arise, I had done a postdoc, and when they did arise, there were just, so many brilliant candidates that applied. So I just couldn't get one. So I decided to do a postdoc with Elise, primarily because she was working on NeuroDev, and I made it very clear that I still wanted to work on African populations and with African people based on being a Ginger fellow as well, because I know Ginger is very, very prominently says that that's what they want for the trainees, is to continue to work with Africa. So even if you get an opportunity, it's wonderful. It's a wonderful learning experience to be able to have that opportunity to go and train with brilliant people in different parts of the world. So that's a great thing. But just making sure that you stay honest with the people that you're working with, that you still want to work on African data, you still want to work on these African studies, you still want to have a say in pushing that forward. And it is concerning that you know, we're moving out of the country, and the lack of job opportunities is the biggest concern. But I'm sure with organizations such as PGC Africa, we can do something with funding to improve on that. I mean, if a job opportunity came up for me to go back to South Africa, I would take it. I don't want to leave South Africa. I love South Africa, it's my home, but unfortunately that was my situation. So I think that's why collaboration and relationships are so important across the globe, you know, not just for funding purposes, but just so we can all try and work together to propel African research as best as we can wherever we're situated. So I was very fortunate because I had a great relationship with the NeuroDev team. So make sure you trust the people you're working with as well to have your best interests at heart.
That's so true. Yeah. Niran, as a recent inhabitant of the UK, do you have anything to weigh in here? Do you have some overlapping experiences?
Yeah, I mean, I'm very conscious of the fact that we've run way out of time. But
oh, have we? Oh gosh,
what I would simply say is that what I have found is that there are many ways to be relevant to the continent if we're flexible in our thinking. Wherever we are, if we carry the continent in our hearts, we will find a way to be relevant to what's going on the continent.
Yeah. So I've not been doing a good job as moderator, so apparently we've run out of time. Are there any last questions from people in the audience? No, looks like no. So I'd like to thank again our speakers. It's been a great pleasure to host this meeting on behalf of PGC Africa. And thank you, and see you next time. Bye, everyone.
Thank you, everyone.